Isle of Wight Wildlife Trust. So we'll crack on with the agenda. Let me just get the next slide up. So the main points we want to cover are why is it a good idea to monitor? Then we're going to look at some different surveys, particularly the rapid grassland assessment and some case studies for that method, and then pollinator count surveys, and again some case studies to sort of look at different examples and then thinking about how we might analyse the data. But it is useful to just sort of look at the difference actually between surveys, which are can be a sort of one-off snapshot to discover what species are present and their condition or population, and monitoring, which tends to be more of a long-term approach and is the comparison of repeated surveys to measure change and answer specific questions. And it's that monitoring that can be really useful over time, done in a repeatable way, that can really give us some useful information and data, and particularly things like trends over time. And monitoring is great because it gives us lots of valuable information on things like habitat condition and biodiversity, and that feeds into site management. If you're managing, even a small green space or a verge, doing some regular monitoring can be really useful to give you some information, for example, to help meet objectives or to give you more information about how best to manage the site and the kind of results of management, if you like. And can feed into longer term nature recovery planning, and provide more information to contribute towards local nature recovery strategy, for example. And it's very useful where you are planning or thinking about improvements, and when it comes to grassland, that might be something as simple as relaxed mowing or meadow creation, to do some initial surveys which are kind of give you a baseline, and then the ongoing monitoring can measure those improvements over time. And those, whether it's annual surveys or more regular surveys, it's the comparison between years that can measure change, and that can give you some valuable information about the success of the management, and management can then be adjusted based on solid evidence, and that evidence is useful for any other interested parties or stakeholders. So first we're going to look at the rapid grassland assessment. It is designed to be simple and effective and kind of citizen science, so you don't have to be a botanist to use this method, because it does use indicator species, both positive and negative. So you're looking for the kind of presence of particular species in the flowering season, so they're fairly easy to identify, and we'll come on to this in more detail. But you can also include measurement of physical attributes, and you can include these, as many of these as you want to, or just focus on the indicator species. It is sort of flexible. But if you're interested in the kind of height of the grass or the sward, that's something you can include. If there's scrub encroachment, you can include scrub. But a really useful one I like to use is the percentage cover of wildflowers within the grassland as a useful measure. But you can, and it's best to tailor it to the individual site, so you can select the indicator species to a certain extent that represent your grassland and your objectives, and for the attributes that you want to monitor. And if you then repeat these surveys at least annually, and then you can compare those, you can monitor the change over time. So there's some really good guidance and templates out there we can use. So you might be familiar with Hampshire County Council's Pollinator Pledge. So they've got a toolkit webpage with both guidance for rapid grassland assessment and some templates, and we'll come on to those. I'll show you those in a moment. And there's also really good plant life guidance, which goes into a bit more detail. And there's links there, so we can share these slides as a PDF, so you can get the links when we follow up after the webinar, and the slide information as well, obviously. But as you can see there, the plant life one is how to design and undertake and analyze rapid assessment. So that is useful if you do want to go into a bit more detail in terms of particularly things like analyzing the data. So some useful wildflower ID guides, links there for plant life one, and also you might be familiar with the Field Studies Councils. There's the laminated fold-out charts, which are really quick reference and got some good colour illustrations there. You can reference quite easily. So there's some good sources of information. But there's a few things to think about perhaps before doing the survey. So thinking about the extent of the survey. So you can, if it's a big area, you could divide it up and do more than one survey, or you might have a number of verges and you could do a survey on each verge, for example, and then you can compare the data on different areas. You need to think about the positive and negative indicator species that you want to select for your site, and that could be some of the common wildflower species that we know are good for wildlife and provide good nectar and pollen sources, or caterpillar food plants, or native wildflowers you want to see more of. But there are some good points of reference, like the one I've shown here that's contained in this table within the plant life guidance. And this is a table you can see there for positive indicator species for lowland meadows, and they've got different habitats in there with different indicator species for grassland, which is quite useful. But you can see there are reasonably common, fairly easy to identify species there, like common birdsfoot trefoil, knapweed, meadow vetchling, lady's bedstraw. So you're not likely to have all of those, but that's a good point of reference, and you can come up with your own list to a certain extent. And yeah, identify the physical features that you want to include that are relevant to your site, yeah, you think are worth including. And we don't necessarily have to set targets, but it is quite useful, and I'll give you some examples of targets that help measure the condition, if you like. And a useful benchmark mentioned here is species for species-rich grassland. It has to have more than 15 plant species per square metre. And also more than 30% cover of wildflowers and sedges, but that's excluding some more dominating species like white clover, and also injurious weeds like ragwort, for example. And also less than 10% cover of white clover and perennial ryegrass, because those, again, can negatively impact grassland. So that's a kind of useful benchmark. And as I mentioned, I find the percentage cover of wildflowers is really useful to include, because that's quite a clear-cut measure to achieve that percentage cover of wildflowers. So the methodology in peak flowering season on your site, and yeah, just to say, when you're thinking about indicator species, it's good to do a kind of bit of a baseline survey of the different flowering plants that you've got on your site, and just think about it from that perspective. And probably, yes, to start the process and the monitoring in peak flowering season, which may change a little bit depending on your site, but normally June is quite a good time for things to be in flower, and therefore obviously a lot more easy to identify. So the idea is you would walk a W pattern across your selected area, and you want that W to sort of cover most of the area and be sort of representative. And you then stop at regular intervals as you're walking that W pattern and survey a 1 metre by 1 metre quadrat. So on a small site, you might have fewer stops, and a bigger site, you might have more stops. So it can be flexible. There's not a set number, but if it's a If it's a medium-sized site, then 10 is obviously a nice number and sort of convenient for analysing the data. And in this photo, as you can see, I've just used some bamboo canes cut to 1 metre length, and just use those to mark out the quadrat. There are different ways of doing that. So you can then fill in the form at each stop. So I've left a link, put a link in there, which takes you to the Hampshire County Council Rapid Grassland Assessment recording form, which you can use. And you mark down at each stop the indicator species, both positive and negative, you find in each metre squared. And also the physical attributes like the percentage cover wildflowers, for example, within that metre squared. Then when you're back at base, you can enter that count data onto an Excel spreadsheet. And again, that's provided on the Hampshire County Council Toolkit webpage, and there's a link there. So it's that template for results spreadsheet you can use. And it's important that for that particular verge or particular grassland area, you use the same method each time, so that each plot doesn't have to be in exactly the same location, but just walking roughly the same W pattern, and sticking to the same number of stops would be a good idea, so that it's directly comparable between surveys. So this is an example. This is from the spreadsheet, but the recording form is very similar. Actually, it's the same sort of grid. Yeah, this is copied from the spreadsheet. So you can see across the top 1 to 10. So we did 10 stops of the 1 metre squared in this area, which is a kind of urban wildflower meadow area. So within stop 1, within that 1 metre square, you can see that first attribute I've got there is percentage wildflower cover. So looking at the 1 metre square quadrat, we estimated that 90% of that area of that metre square was wildflowers, which is quite high, which is great. So we've obviously written that down in there, so 90% cover. We then, in this case, just started noting down all the sort of main wildflowers that were flowering that we could easily identify. So in that first plot, as you can see there, we had black knapweed, birdsfoot trefoil, ribwort plantain and dandelion. So we weren't worrying about the number of plants within that metre or anything like that. It's really just a tick to just show that you've got the presence of that particular species in that metre square. And you can see in the bottom of that column in number 1, we've just totaled those 4 species, and that gives us an idea, an immediate indication of the diversity within that metre square of 4 different wildflower species. So effectively, you can sort of build up this list of wildflowers as you go through your plot. So in the next plot, number 2, we had ribwort plantain and dandelion again, but then we added oxeye daisy and salad burnet and yarrow. That you can see there, and we built up the species list, and we added some others like hedge bedstraw and wagwort, viper's bugloss, to give a total of 8 species within that plot. But after considering this survey, we highlighted those in green that we felt we wanted to increase in abundance and were indicative of wildflower meadow habitat. So that excluded hedge bedstraw and ragwort, for example. Ragwort is an injurious weed, so we don't necessarily want to see that increasing too much, although it is an important part of the habitat as well. So you can see we built up this list across the 10 plots, and yes, you can clearly see the total species in each plot across the bottom there, ranging from 8 species in number 2 down to only 3 species of wildflowers in some of the plots. And then in the right-hand column there, at the very end, the totals, you can see the number of plots with the species, which is a measure of abundance. So with black knapweed, you can see that occurred in 4 out of the 10 plots, and so on and so forth. As you go down the list, you're getting a simple measure of abundance of those wildflowers across the grassland, which is really helpful. And that in itself is useful and can be compared between surveys and between years to just compare the abundance and the diversity. So I should say, once you're sort of confident in the peak flowering time, this can just be an annual survey. You don't necessarily need to do it multiple times. Just to be clear, it can just be an annual survey. Then the negative indicator species, as you might expect, are things like thistle and docks, bracken, nettle, bramble, and they've been, we also noted those down for the 10 plots as well, and have an abundance score or total for the negative indicators. So hopefully that's clear. And then I think I've got a different example here where we've added in targets. So this is a different survey on a different site, I should say. But again, we've got the 10 plots, and we've again looked at the percentage wildflower cover in each plot, and you can see we've set a target there of 30% of plots, or in this case, 3 out of 10 plots. We want to have greater than 30% herbs going back to that, or 30% wildflowers going back to that benchmark measure of a good condition grassland. And in this particular survey, you can see that actually 10 out of 10 plots had greater than 30% wildflowers, and so that's a definite pass, and the result is yes, 100% of the plots met that target, so that's a good pass for the condition. Then with the positive indicators, we focused on only including positive indicator species, so we haven't included anything else, so to give us that focus on positive indicators. So they're all green there, species that we recorded. And so it's useful, one target you can use that's commonly used, and again is fairly simple and straightforward, is for 30% of the plots, again in this case 3 out of 10, to have 5 or more positive indicators. So in this particular case, we had 7 out of 10 plots with 5 or more positive indicators, so we've got a result of 70%, and again that's well above the 30% target, so we know that this is sort of meeting our target and is in good condition. Now you don't necessarily have to use that particular target, and you can adjust the targets just to sort of give you a measure and to highlight when it does drop below a target, for example, then you know that perhaps you need to do something about it. And I suppose that there's an example there with the negative indicator species, and the target for thistles is less than 50% of samples should have spear thistle ideally, and in this case the result was 70%, or 7 out of 10 plots had spear thistle or creeping thistle, and that failed to meet that target, and therefore that highlights or indicates that there could be an issue if it's not addressed. and you might want to do some management to kind of reduce the thistles within the grassland. So just another example, I suppose, to see you can do things in different ways, and this is a similar survey to the very first one we looked at, but we've used the Darfur abundance measure to look at the condition and the abundance of each wildflower. So that the score, for example, with black knapweed being in 4 out of 10 stops, we've classed as occasional, and you can see that the scale there on the right, the Darfur scale, which is dominant, abundant, frequent, occasional, and rare. So if a plant occurs in 3 to 4 stops, that's occasional, or 30 to 50% of the plots. And you can build up a kind of abundance measure in that way as an alternative. It's slightly more complicated, but some people might find it interesting and useful to do it that way. And again, you can set targets. So in this particular example, the target for positive indicators was 2 species being frequent and 2 species being occasional. And you can see that highlighted in green on the table there. And the result in this case was 4 occasional and 2 frequent. So the target was achieved. And you can use that Darfur scale again for your negative indicators. So in this case, the target was no more than occasional abundance. And so that met the target because there were no frequent negative indicator species. So yeah, hopefully that's given you an idea of how you can measure grassland condition, which just provides a really useful evidence base, I think, particularly when you might be working with councils or other landowners or land managers to give you that that measure of condition. And again, whether the condition is changing over time, whether it's improving or declining, when you're looking at that year-on-year data. And yeah, it's worth bearing in mind that natural fluctuations do occur. So you really do need to be consistent over time and build up, a reasonable number of years to until you can begin to see any sort of clearer trends. So yeah, just something to be aware of. So yes, you can obviously measure the effect of the site management and to gauge if it's having a positive or a negative effect, and then adjust the management accordingly. For example, changing the time when you might be doing grassland cutting or doing more frequent cutting. For example, if the grassland is not achieving 30% cover of wildflowers, doing more frequent cutting and collecting could begin to reduce the nutrient levels and reduce the grass dominance and increase wildflower coverage. So I'm going to move on to pollinator counts, and I've just got some fun little videos from my garden of a bee hotel, and this is a red mason bee. You can see that's been filling in one of those little holes, and you can see the one on the right there has already been filled in with the eggs and the pollen that they've put into these cavities. And then the other one, these are tree bumblebees that have moved into an old bird box, and I think they've used it before. And if you keep your eyes peeled, you'll see there we go. There's a worker bee with the, and you can more clearly see the white tail and the pollen baskets. They're the ones that go out and about and collect the pollen, obviously, but they're the ones within that you can see that stay within the box are called house bees because they do stay within the nest and help maintain the nest. So pollinator counts, again, it helps us understand biodiversity and provide that evidence base to make decisions and inform site management. It can help you identify both good areas for pollinators and perhaps areas that aren't so good for pollinators that you might want to improve and do something about. So giving you that initial baseline and then continue the monitoring from there. And yeah, particularly if you're trying to improve an area, it can help measure that impact over time. So there's a few different useful sources of information and sort of guidance on the form. So there's the UK Pollinator Monitoring Scheme and their FIT counts or flower insect time counts. So there's information online about those. That they are stationary, so you're just recording pollinating insects that visit one patch of flowers, and you can do those anywhere. And there's a really good app that you can download onto your smartphone and use the app to do a fit count. And there's also a really good ID guide for the pollinating insects within the app. But the one that we're going to focus on is the Hampshire County Council have used the insect groups that were set up as part of the Fit Count scheme by the UK Pollinator Monitoring Scheme and have used those to develop a pollinator count method, which is a kind of transect method where you walk along a set route. And again, on the Pollinator Pledge Toolkit web pages, there's information on there, which I'll come on to and show you more about. So pollinator counts, normally from the 1st of April to the end of September, when temperature exceeds 15 degrees in dry weather. So obviously keeping an eye on the weather and avoiding windy, rainy conditions. But particularly the fit counts, you can do as many as you like. But with the pollinator counts, the transect ones, we recommend to do monthly surveys where you can. That's ideal. But if you can only do one, aim for late spring. But it's important that you repeat them at the same time each year using the same method, so that you can more easily compare results more accurately. So obviously you need to think about the different sites you want to monitor. As I say, you might have a number of verges or a meadow area or a whole park, for example. So if it's a large park, you might divide that up into different zones or different areas. Or a smaller park, you might plan just a walk around the boundary of the site, for example. And it's best to kind of draw up a plan of your route so that can be shared. And, it could be a small group of people doing different counts and joining forces to do those on different months, for example, and just being clear on the route. And just as an example, I've included this image you can see from, just trying to remember, Alton, I think it was, on a site visit recently. And so the community group there have done a fantastic kind of more ornamental garden on a roadside verge. And also on the opposite side of that path, there's more of a meadow wildflower area. So as an example here, you could, a transect might simply be walking along the pathway in the middle there and just looking at the meadow to start with, and just slowly walking along counting the insects as you go, and then repeating that on the other side, on the road side of the ornamental garden, to cover them both in one count. Or if you wanted to, you could do 2 counts, so one for the meadow area and then a separate tally for the ornamental garden area. And obviously that would allow you to compare the two, which might be interesting as they develop over time, to see, which pollinators are using them and if there's much of a difference. Of course, you might get some pollinators using both, but it still might be an interesting comparison. So you've got your route, so you'd walk along the route at a slow pace and count all the pollinators you can easily see. So you don't necessarily need to count every single one. It's more just doing a representative count, because you don't want it to take too long as well. And then you keep a tally on the recording form. I've included a link there, and you can see a shot of the recording form with the different details on there. Obviously, the weather and location, and then those are the insect groups that you are keeping a tally of, and we'll come on to those in a minute. But the important thing is to try and be consistent each time, so that you're obviously following the same route, roughly the same time each month, year on year, I mean, and roughly the same amount of time as well. Is good to keep an eye on the time, just so that it's comparable. So these are the pollinating insect groups, split into 10 groups from the UK Pollinator Monitoring Scheme. And you can download, they've got a really good ID guide, so I've included a link to that. Or via the FitCount app, which I mentioned on a smartphone. So on the smartphone, for example, these are the main groups as are shown on the screen there, and you can click on bumblebees, for example, and it gives you more information on those. But just to give you some quick guidance on those groups right now, and then you can look at that in more detail at your leisure. So obviously you don't need to know what species of bumblebee it is. You just need to know the difference between bumblebees, honeybees, and solitary bees, for example. So bumblebees are generally quite round and very hairy. Solitary bees less round and less hairy. And honeybees are more slender and have very few hairs compared to the other two. And yeah, it will take a little bit of practice to get your eye in, but yeah, you can. Honeybees are pretty distinctive in particular from those other two. Wasps, we've obviously got social wasps, but that's a solitary wasp shown there, and they can be distinguished by their quite large rectangular head and the wasp waist, and they're very, very sort of long and slender. We then got the hoverflies, which are quite distinctive, particularly because of their flight and the way they fly, but also if you look at, you can see in that image the eyes in particular are very large compound eyes which take up most of the head, which is quite distinctive for hoverflies. And then any other fly that isn't a hoverfly gets lumped into that category. We've then got butterflies and moths are recorded as one group. They're pretty distinctive. Then beetles. So one of the characteristics, or some of the characteristics there, are the metallic wing cases to look out for, that have that line down the middle where the wing case is open, as opposed to some of the other insects like shield bugs, for example, have much more leathery wing cases, and they tend to have that X kind of pattern on the wing surfaces. and then any smaller insects under 3 millimeters are in that category. So yeah, there's some useful ID guides that I mentioned that you can refer to, but another within the app, and there's a downloadable ID guide that I mentioned on the website. But this Field Studies Council one is good for reference out in the field, particularly if you do want to. It can be useful to start looking at particular species and making a note of those if you want to. So that's quite a useful reference one, and that's got honeybees and the solitary bees as well on that sheet. So those are only about £4, I think, from Field Studies Council. So you can take your recording form when you get back home. You can enter the counts onto an Excel spreadsheet, and there's a template, and this is a screenshot of the template, and that's again on the Hampshire County Council Toolkit webpage, and you can see what they provided here with the site details, the weather details. Obviously important to have the date and the time, and then all the groups there and the number of bumblebees, for example, that were seen on that particular date. So you can then, there's obviously additional columns there where you can add further counts, and then you can obviously add and sort of change this to how you want. And they've got different tabs for the different spreadsheets you can see along the bottom there for different sites, or you could arrange it by year, for example. And if you build up a lot of data, one thing I've found helpful is to have a summary tab where you can have sort of annual summary data for each year in columns adjacent to each other, to allow easy comparison between years. So this is an example from quite a large park, Hammersmith Park in London, and we decided here to break it down into 3 distinct areas within the park, so that we could see kind of the differences between them. So we had a kind of formal flower bed area with lots of catmint, for example, and that had lots of honeybees, for example, and a reasonable variety of other groups. The meadow and tiny forest and scrub edge area did have more more groups and more abundance in total, so that was quite interesting to see as well. And then around the pond, actually less diversity, but lots of honeybees coming in to take water from the pond, which we didn't get in other areas. And you can see I've also noted down some of the species. You don't have to do that, but if you want to, you can, to just note down some of the species that you've seen. So I've totted up the totals. You can see the diversity with the number of groups at the bottom of each column. And then I've done a total on the right-hand side there in terms of abundance of each group, with a total count of pollinating insects on that date is 120. So that's the summary column. That's a column. The total column is something you could add to a summary sheet, including when you've got counts from different years to compare it to roughly the same time of year. So then just thinking about data analysis, as I've shown, you kind of getting an indication of both abundance and diversity of pollinators, which is a kind of a useful information to improve our understanding. And when comparing year-on-year data, you can hopefully begin to see whether things are increasing or decreasing, or indeed staying the same. And yeah, important to note, like with the grassland, that natural fluctuations do occur, with trends becoming more apparent over a number of years of recording. Trend can be helpful to see if site management is having a positive effect on numbers. So to really help measure those changes, and hopefully if you're making improvements, those numbers might increase. However, it's worth bearing in mind that with the widespread declines that we're experiencing, you know, in the wider landscape, even maintaining stable numbers is a good sign. So I believe that was the last slide. So I hope that's been useful, and we'll stop the recording and take some questions.